Basically, the microscope word has came from Greek word. It is a combination of two words, that is microns means small and scopus means to view or to see. Okay. And the intended purpose of microscope, it is to view or to find some teeny objects which we cannot find through naked eye. Okay. And the total overall process to observe or examine a biological sample through microscope, it is called microscopy. Okay. Then it is a brief history. At first, that Hans and Janssen 1590, they have prepared a complex microscope, a compound microscope or complex microscope. That is, we have different, previously we had different two types of microscope, that is simple and complex microscope. What is simple microscope? Do you have any idea? Only a single lens we have to use. Okay, like a magnifying glass or a simple dissecting microscope. Okay, but later on that research and all those things, the scientists have introduced more lenses to get more magnification and the resolution also. These two words, it is very much necessary for a good microscope, that is magnification and as well as the resolution. Okay. Then Antony van Leeuwenhoek, he has prepared a simple microscope, that is a single lens, and he has found some microorganisms as well as spermatozoa. Later on, Robert Hooke, he has developed a compound microscope, and from that time slowly it has been increased and it has been added that light source or some lens, some changes is there, and nowadays we have, modern days we have basically these three type of microscopy. Okay. That optical microscope, that is mainly light, that use light source. Okay. Visible range of light. Okay. And that have another 5 division, that is Brightfield microscope, Darkfield microscope, phase contrast microscope, fluorescent microscope, confocal microscope, like that. And next we have electron microscope, and that is also 2 types, TEM and SEM. Okay. Then another, it is there, it is basically used for research purpose or any atomic structure to find, that is scanning probe microscopy. Okay. And these are the parts. In the lab you have seen the microscope, that is a compound microscope, compound light microscope only, and we have these parts. Base is there. After that, in that base we have a light source. Okay. That light is visible range. Visible range light means 400 wavelength, it is 400 to 700 nanometer. Okay. And light source may be a tungsten filament or nowadays LED light also, it may be. Or after that we have stage, and below that stage, just below that stage, we have condenser, and condenser have a aperture. Okay. Through that aperture we can control the whole, that total area of the condenser. Okay. And condenser will condense the light. Okay. And condenser will condense the light, right. Then that condensed light will fall onto specimen, because this stage we have, we can put specimen or slide, that light will fall on that, and after, if a specimen, this is a specimen or bacteria, if light will fall on this, then what will happen? Any idea what will happen? That light may get reflected, it may reflect or it may pass through. Okay. And other things, it may get refracted. Okay. And those light we can observe through that is that lens, here some lens is there, that is called objective lens or objective. Okay, through objective another lens is there near with our eye, that is called ocular lens. Okay, through ocular lens we can visualize the object. And some key points, that is through microscope the primary thing is that we have to magnify the object, and as well as we want a better resolution. Without resolution it is nothing, right? Because if we are taking a picture in a mobile, through the mobile, after taking that picture, if we are increasing or if we zoom in, what will happen? After some times the image will get blur, right? Means magnification will make the image bigger, but resolution will make the image sharp, crisp, clear, right? To get a crisp, clear image, the resolution must be higher. Okay, and the resolution obviously it is depend. I will draw this. If these two dots, can you find these two dots, right? Easily you can find through normal eye, but when these two dots is near about here, maybe you cannot find, right, from your side. And this distance between these two dot or two line in different entities is called, what it is called? It is called limit of resolution. Okay, then this thing it is you can find it, right? This is D. Okay, these two dots it is in different entities, and D should be less, then resolution should be high. That will, now I will go through some mathematical calculation. Okay, and means resolution R is inversely proportional with D, that limit of resolution. Okay, slowly I will go and some little bit I will discuss, because it is more about physics and mathematics. Okay, and if R we want more resolution, magnification it is some different part. We can increase by lenses and all, but by the same time we have to increase the resolution also. Otherwise we will not get a clear picture, right? We want a clear picture to observe that cell or cell organelles and all. That's why we need a clear, crisp, clear picture. And this D, again this D, it is equal to lambda, that is wavelength. Okay, lambda, that is wavelength. In physics, lambda, that is wavelength, denoted by wavelength, and we have to take one constant, that is 0.61. Okay, and this is another thing, that is we have to put it NA. What is NA? NA is numerical aperture. Numerical aperture, just arbitrarily I will go through this thing. If this is a specimen, okay, here we have condenser lens, here we have a light source. Okay, from this light source, light will pass through the object and it will go inside the objective lens, right? Here we have, here that this light will go to the condenser lens, and condenser lens will condense this light. Condenser lens will condense this light, and again it will go like this to the objective lens, right? this portion is called numerical aperture. This cone, light will form a cone, right? That cone, it is called numerical aperture. And this numerical aperture, it is depend on another two things, okay? That is refractive index, okay? And another, this here one cone will form, one angle will form, right? This angle is called theta, or we can say that alpha, okay, or alpha. Then again I am coming for refractive index. When light will travel from one medium to another medium, light will get bent, okay. Here you can find that when the light will go from one medium to another medium, light will get bent. That's why in oil immersion lens 100x, that whole of the lens, it is very teeny, and we are increasing this thing to get more light, to get the more resolution or more magnification, more visualization will be more, okay. And that's why we are using immersion oil, and that immersion oil, it is mainly the oil. What is the oil? Cedar Root Oil we will use, right? And the refractive index of Cedar Root Oil, it is 0.51. It is near about 1.51. It is 1.51, okay. And if we want to D lower, then only R will be higher, resolution will be higher, right? Then if we want D lower, this portion should be lower, right? Then wavelength should be lower. First. Second, this again we can write it here, lambda into 0.61 n into sin alpha, okay. This n should be higher, because it is in lower. In the mathematical calculation you can understand this is in the down, right? That's why this thing should be higher, then only this value will be low, and if this value will be low, obviously R will be high. Again I will come to magnification. Here you can find when a light passes through a lens, okay, that this portion, it is called optical center O, okay. And when light will passes through optical center, it will go as it is, but when it will, rather than optical center, if the light will pass, it will deviate, okay, and it will come to the another end of the lens, and it will cross each other, and it will form an image. It will form an image. That image, it is shorter, right? First case, it is shorter. It is shorter image, and it is a real image and inverted, right? Real image and inverted. But if we will bring the lens or the object, if we will bring close lens or lens to the object or object to the lens, then what will happen? Then the second case will, it will be the second case, that image will form, but it is also, it is some magnified and it is inverted real image. But if we make it more closer, it is near about that opposite to the focus point, then what will happen? Then that another image will form, that is not a true image, right? Okay, and it will form behind that object. Okay, and this principle we will use in the microscope, what we are finding. Okay, this is here H0 is forming H dash here, that image, it is inverted image but real image, and this image will form, this image again will magnified here, and this is virtual image, and this image Hi, it is the image of H dash. Okay, and now the thing it is about bright field microscopy. It is a part of light microscopy what we have in that in our lab. Basically the field it is bright, and the object it is dark, because we are staining the object to make it dark. Okay, to make it some little bit contrast, right? That's why here you find that particular image, how it is forming image, it is that particular object or bacteria, it will put some color, will stain the slide, and after that only we can find. Okay. Next, this is dark field microscopy. Actually dark field microscopy, use of dark field microscopy for the living organisms. If we want to find some living organisms, we have to use dark field microscopy. Okay, any movement of any organism or any extracellular or any parts like flagella and all, we have to use dark field microscopy. Here dark field microscopy, it is a kind of light microscopy, but we have one annular ring over here. Below the condenser we have an annular ring below the condenser. In bright field microscope we don't have any annular ring below the condenser. Okay, but below the condenser we have an annular ring, and that annular ring will pass the light in the from the periphery region. Periphery region the light will pass. Okay, and if periphery region light will pass, what will happen? If a light will pass through a microorganism, what will, if suppose this is a microorganism, bacteria, okay, and light will pass, okay, and light will pass like this, but after passing the microorganism, the wavelength will be short, okay, and this will make the contrast, right, and whatever the diffracted light we will find through objective lens, okay, and the background will be dark like this. Organism will be bright and the surrounding field will be dark. Yes, annular ring here, this is the annular ring. This is the longitudinal section of the dark field microscope, right? This is longitudinal section, okay, and this is cross section of the annular ring, okay. Here it will be, this portion will be, light will be closed, and from light will pass through this periphery region, okay, in dark field microscopy, and like some bacteria or movement of bacteria we can find through this dark field microscopy. Here only we are not stable, we want more clear picture, right, more clear picture to find some extracellular apparatus or any other organelles like flagella, cilia, like that. We want a best resolution, best background, like that. That's why we have developed phase contrast microscope. The difference between phase contrast and dark field microscope, it is here phase contrast means here one phase ring is there only, and we have annular ring also. We have annular ring as well as phase ring, and phase ring it is fixed with where? The phase plate will be in the objective, okay, in the objective or in between objective and ocular lens, okay, and here the light will pass through the periphery region, okay, then light will get refracted light will pass through objective, and again in that objective, that which light is not refracted, that will also enter into the objective, and that will be blocked by phase ring, and again we will get more black background and more brighter the image, okay, because if this is wavelength, suppose this is wavelength, how it is from here to here, it is what, lambda, right, and this is lambda by 2, okay, and this is lambda by 4, like that. It will slow the light and the background will be more black and the specimen will be more bright, and this is the picture you can find how we will find the extracellular cilia and flagella and all, okay. Next, fluorescence microscope. Here we will use some short wavelength of light, okay, yes, like this. If energy and wavelength, it has some relation, okay, energy and wavelength we have some relation, because energy is equal to E one by, or you can see it is inversely proportional to wavelength. If energy is high, wavelength will be short. If energy is less, wavelength will be large. And fluorescence microscopy will work like this. Here we have electron, right? E. If UV light will fall on it, it will, that fluorophore is there. We are using fluorophore as dye, like, or I mean like that. And after receiving UV light, what will happen? This electron will get excited, and this excited electron will jump into the higher energy. Energy will get high and it will jump to the higher energy. Here it will come, okay, and after coming here it will not be stable. Again this electron wants to come back to his energy level, okay, and that time when it will come here, that time it will release some light, okay, that is fluorescence, okay, that is the fluorescence. And this is the particular microscope. Through this microscope we can take the image through computer, we can visualize through our ocular lens also, okay, we have camera also. And how this kind of microscope, we have one kind of filter, ठीक है, तो that is called, or mirror also, we have some different type of mirror and some different types of filter is there. Those mirror and filter will pass a particular type of wavelength. Okay, we have excitation mirror, excitation filter. Through that excitation filter, what will happen? Only which kind of wavelength we want to pass through, that we will adjust. Okay, and we have a dichroic mirror. Through this dichroic mirror, that whatever we want to pass through that reflection, we can put it on the objective, okay, or object, and from the object it will get reflect, and from the dichroic mirror it will pass through, and it will pass through emission filter. That emission filter only filter that particular range of light. Okay, when it will reflect some other also it may come, that is out of focus. Okay, those some light will be out of focus, those light also will be filtered by this emission filter, and it will captured by the camera or our through the ocular into our eyes. Okay, and to get basically use for pathogen detection, Microbacterium tuberculosis, where we use generally we use Oramine to glow that particular bacteria, and it will grow in the slide, that is yellow color will come. Okay, again confocal microscopy, to make more particular, or to make the pinpoint focus from the fluorescent microscope. Again another division is there, that is confocal microscope. It will, the focus portion will be more in this confocal microscope, and through this we can, and we have a different kind of filter here. Dichroic mirror is there, but we have a pinhole filter, and through that pinhole filter we will use laser over here, laser light, and that laser light will pass through that pinhole filter, and that too much focus light will fall onto the specimen, and specimen will reflect that light, and that light will pass through dichroic mirror, and again it will fall on the pinhole filter, and that pinhole filter will cut off all the out of focus light, and only the focused light will go to the detector and will find that particular image. That image will be like that, and by increasing the intensity of laser beam, we can find different area or different portion like mitochondria and all in a cell. We can find these things in a very crisp, clear picture. And this is the confocal microscope is like this. Okay. Next it is coming electron microscope. Electron microscope, it is transmission electron microscope, and another it is scanning electron microscope. Transmission means it will pass the light through the specimen. Okay, and scanning electron, whatever the 3D structure of the specimen, that will find it, and we can measure that structure, outer structure, and how it is. I will go through that image. You can find that particular image in scanning electron microscopy. Whatever that 3D structure, we can find it, but through transmission we can find a inner side structure, and sample preparation also it's different. We have to cut the sample in transmission electron microscopy in a very fine microscopic particle, like we have to use not in different types of knives, we have to use diamond knives to cut the sample, and we can find that inner side of the inner structure of that particular organism. And here also you can find that fluorescent transmission electron microscope, we have a fluorescent skin. On that skin, that image will capture, and that will direct to the detector. Through detector, and computer system will be there, software will be there, through that we can find a better image. And the same thing for scanning electron microscopy. Here we are in this two only. Electron microscopy means we are not using light, we are using electron, right. And again the same thing, when electron will fall on some electron, high intensity electron again it will cut out that particular electron, and that electron will be captured by, that is called secondary electron, and that electron will be captured by the detector and the 3D image will form. Okay. Then next it is atomic force microscopy. This is means a vast portion of microscopy, and here a particular nanometer scale we can find, okay, through this microscope, and different type of structure, 3D structure we can find, even some bond in a protein, how it is forming, particular amino acid, distance of that particular amino acids, like that we can find through atomic level, also we can find through this microscope. Okay. These are all about your microscopy. If you have any doubt, you can. Hopefully you got that some little bit portion of microscopy, and that will, I think, that light microscopy that will help you to focus and how it is working, resolution, magnification. Okay, magnification is a part, but resolution is the main, and on that resolution only the value of a microscope will decide.